This is Emily Vinolo. Thanks, Anna, for the introduction, and thank you all for attending this webinar. We're happy with Enzo Medico, the coordinator of the EurOPDX research infrastructure, to present you today an update on the IT tools we are developing to support cancer research with patient-derived xenograft models. So this is a progress this is presenting our progress as compared to the first webinar we hosted with the EBI training about a year ago in October 2019. And but before we go into the demos of the IT tools, I will first introduce you briefly to the EurOPDX research infrastructure and the opportunity to access the resources of the infrastructure. So briefly, some of you may not be fully familiar with patient-derived xenograft models that are established by implanting a tumor fragment or a biopsy from a patient in an immunodeficient mouse. The tumors grows in the mouse with a relevant, with a particular success rate, and upon reaching a certain size, a maximal size, the mouse is sacrificed, and the tumor can be reimplanted in a second generation of mice or second passage. This can continue over several passages. At each generation, the tissue is biobanked as formalin-fixed, paraffin-embedded, fresh frozen or cryopreserved, and a certain number of validations and characterizations are performed. Those include histology, fingerprinting to track the genealogy of the samples, sequencing of different forms, and drug dosing studies. So this allows to validate the PDX model, to compare it to the original tumor of the patient, and of course to characterize the tumor. So it's been shown widely that the PDX models keep the main feature of the original tumor of the patient, and they are now widely recognized as clinically relevant preclinical models. But they are also time-consuming and costly to establish and to characterize to be able to use them optimally. So several years ago, centers, laboratories in the EurOPDX consortium decided to create this network to integrate collections of existing PDX models. That would allow to perform studies for genotype response correlation on a population scale by tapping into a wider collection, distributed collection. And of course, in doing so, increase the predictability of preclinical data for success in the clinic, since we would be doing drug dosing study in a population representative of the heterogeneity in patients, and therefore improve oncology development success rates. So that was in 2013. So the initiative was to promote scientific collaboration in PDX research, integrate the collection, as I said, and raise standards in the field. And a few years later, with all the key partners, we were awarded a 4-year grant from the EU. And this grant is to build a cutting-edge and novel EurOPDX research infrastructure. So this is to support PDX research in Europe, but also worldwide, and in particular by opening access to the PDX models, expertise, and resources of the consortium for academic researchers, but also for industry. So those are the main objectives of the project, of the grant. So one of them that we will be focusing on today is to develop and employ efficient and user-friendly IT tools. So capture the information for partners on the models and their annotation, integrate this data into a virtual repository. This is our EurOPDX data portal, and enable the searching and an analysis and the visualization of this data for model selection, but also for hypothesis generation. As I said, we have a big focus also on standards that are needed in the field, but I won't focus that much on this today. Another thing I would like to present to you is how you can benefit from free of charge access to the models that we call transnational access. So I'll go through the details. Again, about standards, I was presenting a little more at the first webinar last year, so the video is available on our YouTube channel. And otherwise, we have a standards tab in the webpage, in the EurOPDX website, and we'll be publishing these standards as well. So I invite you to visit those material if you are interested. So what is transnational access? So this is a way through the grant to provide free of charge access to PDX resource, again, not only to academic labs, but also to industry in Europe and worldwide. And this access to models is preferred through 6 state-of-the-art partner facilities that we call nodes, and they are depicted in the map on the right. And the reason we are also hosting this webinar is that the last opportunity to apply for transnational access under this grant is approaching. I mean, the deadline is approaching, and it's on the 18th of September. So it's on Friday next week. And the application process is pretty simple and straightforward. So there is still time to consider applying. There are 3 ways you can access the models or the data of the research infrastructure. The first one is called sample. So it means that we would ship frozen or viable PDX tumor samples to selected academic laboratories. This is for academic labs only. So after selecting the model, your model of interest or your models of interest in the data portal, we would expand the model according to our standard operating procedures and do the quality control according to our SOPs as well. And finally, ship the models to you together with a PDX passport, which is detailing for each model the main characteristics and the quality controls. Another way to benefit from our research infrastructure is to deposit the models you have established yourself into the biobank and the data portal. So we would perform a sample health check to verify that the samples can enter one of the animal facilities of the nodes. We would again expand the models according to our SOPs and do the quality controls, and store them in one of the biobanks of the one of the 6 nodes, and publish the model of the on the EurOPDX data portal. So this would promote visibility for your models and collaborations with third parties. Finally, the test access is also available for industry, and it allows performance of in vivo drug efficacy studies on selected models. So in that case, the user of the research infrastructure would provide us with the drug or the drugs of interest, and we would perform the in vivo experiments, again, according to our SOPs, and deliver at the end of the project the experimental results and the quality controls. So again, you can apply for access by 18th of September. All the call info and application documents are available on our website on a dedicated TA or transnational access webpage. And if you have any question or you can't find the material, please contact us through the help desk at ta@europdx.eu. So we run 3 different calls already in the grant, and the projects are with some delay due to the COVID situation in particular. Those are all being initiated and will be completed in the next month. So in terms of IT tools, all this started with the PDX Minimal Information Standard that was published, or PDX-MI, that was published in Cancer Research in 2017. And this was under the leadership of Emboli BI and with the EurOPDX members, but also the wider PDX community with US labs, etc. There was an agreement on the minimal information that need to be available on a PDX model to make it a reliable model. And so thanks to this, the EBI and the Jackson Laboratory developed the PDX Finder that is powering our EurOPDX data portal. So you have the link to the data portal here. dataportal.EurOPDX.eu. The latest release was at the end of July, early August this year. And so in addition to the Finder, the portal includes additional functionalities, which include a user authentication service to provide user dashboards and track some user details for reporting purposes for us to the European Commission. We are also linking to advanced analysis tools, which are the well-known cBioportal and the Genome Cruiser for 3D visualization. So Enzo will provide you with a demo of the cBioportal and Genome Cruiser. And also you can filter the models for aspects that are specific to the research infrastructure, and in particular the possibility to access them through transnational access. So this is just a picture of the EurOPDX instance of the cBioportal. We have more data than last year included in the cBioportal, but again, Enzo will show you the details. And Genome Cruiser is a tool developed by an SME in Italy called Cairo 3D. It's a software application based on real-time 3D technology to perform data visualization and analysis. So you can really navigate the data in a 3D environment and explore many genes in pathways, gene families, studies, sample clusters. So Enzo will show you all that. So before I go to the data portal to show you some basic features of it, I just want to give you an outline of the PDX collection and associated data that are currently included in the repository. So upon the latest data release this summer, we have currently almost 1,000 PDX models from 8 different research centers and different, even more research teams, among which a little more than 850 are available for transnational access. So the others are only available on a collaborative basis. That means by contacting the owners of the models directly. And for these models, all the metadata are available according to the PDX-MI standard, in addition to gene mutation, cytogenetics, patient treatment, copy number alteration, expression data, and drug dosing data whenever available. And also, of course, the ideal is to build with this repository a pharmacogenomics space. So we will be adding more data as they become available. So with this, before I let Enzo make the demo for cBioportal and Genome Cruiser, I hope you still see. So we are here on the EurOPDX website, so EurOPDX.eu, and this is where you can find all information on the research infrastructure, like transnational access, the standards, and also the PDX collection. So more widely, all the PDX collection, we haven't included everything in the data portal yet, also because it has to go through the PDX-MI filter. And through this page, you can always find a way to access the data portal. So I'm now on the data portal. You can see the number of models that are currently available. And so to search the models, you can either go to Explore the EurOPDX data or the Search tab above. Of course, you have a number of additional tabs to support you in the registration process, explain further the type of access to the models, detail the PDX owners, and more details about the project and contacts. So as I mentioned, there is a need for authentifying to the EurOPDX data portal. So upon registration, I mean, you will have to register first and then log in each time you go back to the data portal. And this is made easier by the fact that you can use your credentials from Google, LinkedIn, or ORCID. And this will be even further developed with institutional credential soon. So I will log in with my Google account. Yeah. And so I'm here on the search page of the data portal. Like I can currently see all the 995 models. So on the left of the data portal, you have all the different filters that you can use, all the different ways that you can filter the data to find the appropriate models for your research. So by the type of data set that is available on this PDX, maybe by the data source, so the laboratory that has established the models to start with. The project is always EurOPDX, and the access modalities. So for instance, if you want to apply to the transnational access call, you may want to click those two filters first, some of the models being only available for in vivo efficacy studies in the nodes. Of course, we can always support you with all of this if needed through the help desk. You can also search available molecular data for a particular gene mutation, a copy number alteration in a particular gene, gene expression. If you are working in breast cancer, looking at breast cancer biomarkers, so the three famous ones, to select for HER2 positive or triple negative, for instance, and also cytogenetics, so immunohistochemistry. You could also look for particular patient treatment status, a particular patient treatment, a particular drug that has been tested in the PDX models, and finally by tumor type, cancer by system, tumor type, primary, metastatic, etc., by sex or by age of the patient from which the sample was used to establish the PDX model. So with this, you have, as I said, different ways to search for a model, and I can make really just a quick search, for instance, for breast cancer models to show you. But this is just a really simple example. So I usually type the cancer type here rather than going further down. So if you type for breast cancer, you see that we have, like in the picture earlier, 88 models of breast cancer from 6 different labs across the EurOPDX consortium. And I can say that I want to focus on HER2 positive breast cancer. So from those 88, 18 of them would be HER2 positive. So from the University of Cambridge, Manchester, Institut Curie, or the Vall d'Hebron in Barcelona. And I can also say, okay, so from these tumors, I would really need that they are mutated in p53. I could select for a particular variant, but if I pick all of them and I search, then it would retrieve one model. So that is one model available from the University of Manchester of a primary tumor. So then you have several information. So a related publication when available, how the model was established, information on the patient, and the molecular data. So this is where the mutation data came from. So you have also some immunohistochemistry, unfortunately not drug dosing study on that model. So with this, I think I can hand over to you, Enzo, for the CBio portal and Genome Cruiser demos, please.
So I'm starting again from the EurOPDX website here, where you can actually find the path to the data portal. You go to PDX collection and the data portal. Now, in the data portal, Emily has just shown how to explore the data and enter the PDX Finder platform for selecting models based on many different things. I'm showing now an additional tool, which is the CBio portal. So data for a large fraction of the models has been ported on this system. So you just click here, and an instance of CBio portal, the memorial sloan Kettering tool for molecular data visualization, has been implemented by Masaryk University, and you can see the studies here: colorectal adenocarcinoma, invasive breast carcinoma, esophagus and non-small cell lung cancer, with a number of samples. Clearly, if you, for example, want to see the study summary, like which you go click here in this pie chart, and you will see something similar to what is provided through the data portal, a summary of molecular, plus a summary of the molecular information, so real molecular data on not just on few genes, but on the onward gene study in terms of mutations, in terms of copy number alterations. There is, if you add the chart, you see this is clinical features of the models, genomic features of the models, and you can also generate custom groups. Now, if we go back to the main page, we can select all the samples, okay, all the studies. Now, in this case, it's almost 800 samples. The profiles that are available for these samples are mutation and copy number alterations. The overlap is not perfect, but it's very large. And imagine you want to select a series of genes you're interested in, not just one gene or one annotation. Let me make an example. You want to find, for example, I have a problem with the window. This is not opening. Just a moment. So you want to find, for example, PI3 kinase family genes, and find models. You are studying PI3 kinase family pathway. And for example, this is very simple. You go on Wikipedia and you find a list of genes. Okay, you're interested in this list of genes, all the various catalytic, regulatory form, and different classes of kinases. You generate a list, like I did here in Excel. What you need is official gene symbols. You can see this list here, and you just copy the list and paste it down here. So now we are exploring a list of genes of interest of the user. And there is a check. Now we submit the query, and now we are opening a view of the data, which is called OncoPrint, where you have for every gene a lot of all the samples here and annotations, colored annotations that provide information on the type of alterations. For example, the blue here and down here means deletion. The greens means a non-synonymous mutation. So every column is a sample. You can see the study of origin. So this is IRCC gastric cancer. This is breast cancer of Manchester. This is colorectal cancer of Candiolo, and so on. So in this case, for example, you can see overlapping mutations for one of plus PI3 kinase family genes. You can go, for example, to an analysis which is called mutual exclusivity, just to see how these genes can tend to co-occur. And in some cases, there's a tendency to co-occur, for example, for different subtypes of PI3 kinases. So you can try to find out in the models that have a co-occurrence, or you can explore mutations gene by gene. So this is the spectrum of mutation in PIK3CA that is available in the PDX model. You can see many different sites, and this is the sample ID, the type of tumor that has the model. So this is a pan-cancer analysis of PI3 kinase. If you're interested, for example, you can see many of them are in a common here. This is very common in colorectal cancer, for example. but not only. You can see there is also one in breast. So if you're interested in studying this, you can test this one and then all the different types of kinases. You can check, for example, if you go back to OncoPrint, for example, you can see there are models that have the regulatory, so P85, PIK3R1 is the P85 regulatory subunit, which is frequently deleted. So you can just check and say, well, but there are models that carry this deletion. Is there anything interesting about that? And actually, it turns out that it's actually a tumor suppressor. If you go here, you can see here, for example, P85 alpha can be a tumor suppressor. So maybe you will find models of interest for exploring this particular way of affecting PI3 kinase pathway. Expression data are still not available. We will put them. We are planning to put them. But you can check for every sample. And again, if you are interested, for example, in PIK3CA, and you want to see the models that have this one, you can check the model, the sample ID, for example, this guy here, you click it, and you go again through the CBio portal, you see all the mutations that were available, not only PIK3CA, but there is also a P10 mutation, so related again to the PI3 kinase pathway, plus a number of other mutations. This is the spectrum of molecular alteration of the specific model, which is this one. So this is the patient, and this is the information of the patient. This is more or less a glimpse of what you can do. PIK3R1, for example, this is PIK3R1. And in this case, you can see there are mutations at different levels and not only deletions. There are also nonsense mutations. You can see nonsense mutations. So there are two ways by which the gene can be inactivated as a tumor suppressor. And there are models. So this is not like the conventional TCGR when you can do nothing. You're interested in this mutation. Here is the mutation, and this is the sample. So you can make experiments on the sample that has that mutation. And these samples has many, many mutations. These are hypermutant, so MSI hypermutant samples, for example. But here's the PIK3CA mutation again. When you go to the patient, you can look at the clinical information that is available and the institution. So this is similar to the data portal, this part, but this shows a connection between the molecular data and the available model that has those molecular alterations of interest. Now I think we have to move to the next part. So this is one of the ways. I remind you that we already provided some guidelines to the CBio portal of the EurOPDX infrastructure in a previous webinar, which is available as a link also in the presentation. So in that presentation, I provide more introductory aspects of the CBio portal. Now I would like to move to the next tool, which is different. This is meant to explore not just one or few genes, not one or few samples, but all the samples together. It is a new software developed by Kairos 3D. It's called Genome Cruiser. I'm just opening now. You have to download it and install it. And in the slides, there are links to email to obtain this software, which is available. So there is a database with the data. We connect to the database, which is here. And there is kind of a scene to open, so a scenario to open. In this case, this is colorectal cancer. And let me show you what we are going to see now. This includes gene expression and copy number. So this is dedicated to gene expression and copy number. So what we are seeing here is a list of genes which overall comprises the kinome. and a list of samples, which is here. You can see these are 43 samples, so 43 models with gene expression, and these are kinase clusters expressed differently across the different models, and these are model clusters based on the kinome. So you can see there are groups of models that express these genes and these genes, and less this family of kinases, and this group expresses this family of kinases, for example. Now, an important aspect is, if you're willing to search for, you're interested in kinases and you want to work on kinase pathways of various types, we also need to know how the copy number values are changed. So here comes the interactive 3D environment. So we move now by interacting, you can see the mouse, we now see the third dimension, and this is going to be the height. So the copy number changes are encoded as height of the single events. So you can see here is a single gene where you have a value for color, which is gene expression, positive, upregulated, and upregulated also for copy number. So in this way, we are seeing in a single view both copy number and gene expression. Now here we are using absolute values, so even gene deletions go up. But if we remove absolute values, now we can see that all the genes that are amplified tend to be overexpressed, while the genes that are deleted tend to be underexpressed. So there is a very good correlation. And you can see that there are avenues where you have recurrent genes recurrently amplified and overexpressed across samples. So this is very interesting. You can be willing to browse through this scenario. For example, let's remove the genes that are not interesting. So the genes that are not amplified or deleted above a certain threshold. So we start removing down and then up. So we clear it up. So we only stay with the genes that change a lot, okay? Like here, there are kinases that are really strongly amplified and overexpressed. This one comes to my mind, for example, Wink1, okay? So Wink1 is, you can select and see, okay, there is a couple of samples that are Wink1, 3 samples that are Wink1 amplified, for example, and there is also something else. I mean, this one here, MAP kinase 7. So again, so MAP kinase family member. Also, you can see more samples that have MAP kinase 7 amplified and overexpressed. So in a single shot, we are browsing all these gene family, so kinases, which have copy number alterations and copy number deletions. So we can flip. So now we see, for example, the strong deletion here of MAP2K3 and a strong deletion also of PANK1, so another kinase. So there are also kinases that are deleted in some samples. So overall, this allows you to have a one-shot, and if you have a gene of interest, okay, so imagine you want to see, as I mentioned, one gene like Wink1, search in gene, okay, select item, search, and here is Wink1 highlighted, so you can see your gene of interest. If it is a kinase, of course, you will see it there, and see how it is altered across the samples, like this. 1, 2, and 3. Whoops. Just a moment. I need to zoom out. Okay. Now let's move now to another way of seeing the data, which is called analysis mode. You can see it here by clicking this analysis mode. This is a different way of showing the data. Now in the floor, now we have all the kinases grouped by clusters. This has been done by clustering. Kinase cluster 1, 5, 3, 2, and 1, 2, 3, 4, 5. And as I mentioned, the samples are now clustered based on their kinase expression profiles. So if I click, for example, kinase cluster 1, And I go on, for example, gene expression here. You can choose between gene expression of copy number variation, you can see. Now I can see that samples of these clusters have this pattern. They tend to express these three clusters and less the others, but not really clear, okay? This one has a different expression pattern. This other has a different expression pattern. This one also, again, a different, and this one, for example, this one, so cluster number 4 of the patients, of the PDX, tends to express high levels of the cluster number 5 of kinases. Now, let's see if this may also be due to amplification. So remember that we have overexpression here. Is there any gene that is also amplified? So we go to copy number, and now the height represents the p-value of the differential statistics, okay? So you can see there is one gene, which is REOC3, rheokinase 3, which is statistically significantly more amplified in this family of patients. So let's look at this kinase. And you can see now here in the color, you can see that there are samples that tend to have copy number gain or amplification in this group of patients, not in the others. But there are these two samples that are particularly interesting. So we can use a pin function. We can pin them out, like here and here. And let's see if they are also overexpressed, not only amplified, but also overexpressed. Now we have pinned them, and you can see that one of the two is not so overexpressed, but the other is. So this sample here is overexpressing and also having amplified rheokinase 3. Now, if we select one single sample, go back to select one single sample. Now we see the complete gene expression profile of this sample. And let me show you what this other panel means. These are gene groups. So every tile here is a list of genes. And for example, this is TNF alpha signaling. So this is a cluster of 157 genes associated with that. Of course, part of them will be part of the kinases, not all of them. And they tend to be overexpressed by this sample. Same with other pathways, interferon alpha, inflammatory response, and so on. These are colorectal cancer subtypes. This is epithelial mesenchymal transitions, which is under expressed. So this will be a more epithelial tumor. We can move to the copy number. And now, very interestingly, we see not only rheokinase. So this is not the only amplified gene that this sample has, but there are also like LIN and another one here, PRD1 and RIP kinase 2 and so on. So it is very easy to get a complete overview of a sample in terms, for example, of kinases. Now, to conclude, because we are approaching to the end, I will show you a very powerful feature here. You can change what you are seeing, okay? So let's, for example, change the genes that we are working on. We change the clustering, and the row clustering, which means the genes, instead of kinase, we choose genomic landscape. So let's see all the genes now. Okay. Now we are looking at all the genes. This is the entire genome, okay? Let me select the sample to show you. If I click the same sample of before, now you can see single genes right here, and this is the entire chromosome 1, this is chromosome 2, again 2, this is 8, 8p, 8q, so short and long arm and so on. So this is a complete genomic landscape of the sample, and you can see that we have this is chromosome 18, which is consistently rather amplified, or it is there are multiple copies of the entire chromosome, apart maybe from this small section. Same from here. So it's a polyploidy of the same chromosome. It is very interesting then to see whether this amplification and deletion are mirrored by overexpression or underexpression. So let's try to pin, for example, just to remember it, this guy here and this one here, okay? And now let's move to gene expression. Now you can see that many of the genes in this chromosome are also overexpressed. And here a little bit less, okay? But clearly this sample has a chromosome 18, multiple copies and many, many overexpressed genes. Now, let's clear the pinning, okay? So where is, we stumbled on that particular sample because of rheokinase, remember? So let's look at here, we have all the panel here. Let's search for, what was the name? Rheokinase 3, I think. Rheoc 3. Genes. Okay, search. Where is rheokinase? Here. It is exactly on chromosome 18, arm Q. So it's part of this. If we convert to selection the pinning, now we will see the copy number here very high in this particular sample. So this is a sample which is very good for exploring chromosome 18 amplifications, rheokinase, maybe other targets. This is not something that you do quickly, okay? I'm now kind of showing one example, but it's very easy to spend half a day or even the entire day browsing into the data, trying to understand what are the real genomic features of a single sample. The possibility of seeing the entire genome of a model, you can see they are different, okay? Look at this model here versus this model versus this model. The genomic landscape of copy number is completely different. If you move to gene expression, same. You can see the geographic allocation of of gene expression domains. Again, you can see very, very interesting how they look different from each other, okay? With this, I think I am finished. It's yeah, it's 45 minutes from after 4. Clearly, I'm open to give questions, and there is a slide. I think Emily has it at the end of her deck with the emails, email contacts. I can just have you. Okay, these are the contacts for the Genome Cruiser. In particular, people from Kairos 3D, Luca Bezzadini and Riccardo Corsi, this is their email address. And then myself and Claudio Isella, who heavily worked on making all this data system, this data kind of parsing and organization possible. So you can write to each of us, any of us, and we will really happy to collaborate, to provide the software, and to discuss further implementation. There are different ways to cluster the sample, different ways to select genes for analysis, and we really want to make this tool useful for people in the PDX community and in the genomics community. So that's it for now.
Thanks, and so I can also put just the last slide with you know, a reminder about the deadline, the links to the call info and documents. So we Anna, thank you for posting the link to the YouTube link to the first webinar. We have a EurOPDX research infrastructure channel, so you can look for the video for the second webinar will be available as well, and there is a video of presentation of the research infrastructure. And again, for any question or feedback, please don't hesitate to contact us, and you can follow us on Twitter as well.